Hello and welcome back to our last lesson in Plant Science 1. It's a big one. Cover two important aspects of horticulture in terms of how plants are reproducing. And so we're going to cover both sexual and asexual reproduction today. So we'll start off with sexual reproduction, which is the fancy term, right? Where you got your male gametophyte and your female gametophyte that are united to form a zygote, fertilization, right? Um, we did talk about mitosis and meiosis a while ago. Hopefully, you remember some of that. Um, in sexual reproduction, this becomes a really important aspect because I think at that time we talked about it. But remember, if you are combining two propagules together, two male and female gametophytes together, they each have their DNA component, and that's great. When you combine, we end up with an offspring, but we can't just keep adding DNA each generation, right? So we have to go through meiosis so that we have haploid. So any part of the flower, whether it's the female or the male, the reproductive gametophyte will have gone through haploid. So it has half as many chromosomes so that when they're recombined, two halves make a whole. So you end up with a diploid offspring. Now, it could be tetraploid, right? These remember the prefixes I talked about on that lesson. So if you take two diploid plants and you combine them, you end up with a tetraploid. So, anyways, ploidy counts with DNA is something that we won't get into too much more in our program, but at least you should know that gametes have gone through meiosis and they have half as much DNA. So, sexual reproduction is really important for plants because it allows for their genetics to be mixed, right? Not only are they mixing by cross-pollinating, but in the process of making gametophytes, some of the genes are allowed to shuffle. So sometimes more so in some species than others. But the whole act of going through sexual reproduction is really important for the long-term viability of any species, whether it's a plant or an animal. Um, so there's the opportunity to introduce new genetic material, right, that might have desirable traits. Um, and pollination is, a big part of it. You're probably familiar with bees and their role and all the insects in the world, but pollination is a really important aspect for that genetic mixing. And pollen themselves are just fascinating. There's some links in the folders for this week for you to go look at, TED Talks and stuff like that. But I don't want to get too lost in the weeds there. Now, when pollen lands on a plant, there are some biochemical processes that are quite complex that allow a flower to determine if it's the correct pollen or not. So you can't just willy-nilly throw pollen on anything and expect it to be compatible. There is some sort of a molecular mechanism that says, hey, this is pollen from my own species. I will let it germinate. That could be the structural interaction of the pollen or chemicals on the exterior. It's not fully understood and it's beyond the scope of this course. But pollen recognition is where it all starts. Now, who receives the pollen? Well, that's typically the stigma. So let's get into a couple of things in pollination. There's two avenues. We have self-pollination and we have cross-pollination. So self-pollination is a transfer of pollen from the anther to the stigma of within the same actual flower or between flowers in the same plant so that there's no genetic mixing really happening. It's self-pollinating, or we call it selfing. Cross-pollinating, opposite to that, means that pollen is being transferred from the anther of a different plant's flower from a different location, a separate plant, onto the flower. And obviously they have to be the same species. So advantages for self-pollination, well, the plants can easily spread beyond the range of pollinators, right? So if you're confined to where a necessary insect is that is involved in pollination, you can't really expand your boundary beyond the movements of that pollinator. Whereas when you're self-pollinating, you can spread all over the place. And grasses are a really good example of a self-pollinating plant that spreads wildly all over the place. Um, They don't have to attract the pollinator, which also means they save energy, right? They're not expending energy on showy flower structures or nectaries in the flowers. Some disadvantages: plants may lose genetic diversity, right? So the fancy term is vigor, but they could be weakened. Or, for example, the climate might change and their particular genetic makeup may no longer be advantageous. Um, so it can also be difficult to introduce new traits, right? So now, in some cases, as I alluded to earlier, self-pollinating plants can vary their own genetics through interesting mechanisms beyond the scope of this course. But so they're not entirely hopeless at creating their own genetic diversity. With cross-pollinating, the advantages are genetic diversity. All right, we've talked a lot about that already, which can lead to two things: beneficial traits or what we call deleterious traits. It doesn't actually guarantee that it's going to be good. It just means that change is going to happen. Um, but we often, the association is that, okay, with cross pollination, there's a better chance that we'll derive some sort of new resistance to a pest or a disease just from the random shuffling of genes. And then whichever plant has the most advantageous traits will eventually become dominant in that particular geographic location. From a practical or agricultural perspective, cross pollination often leads to higher yields. You can see how that would be important. Some disadvantages: well, they require pollinators to vector the pollen from flower to flower. That means that they have to expend energy in attracting the pollinators through a flower or pheromones or nectaries. Um, and of course, I alluded to this. You can, by cross pollination, you could also introduce less desirable traits. Um, Methods of pollination, there are typically mechanical in nature, right? So wind is the classic example where the wind is picking up the pollen and moving it along. This is usually confined to pollens that are very small. I personally have grass allergies, which are a pain in the nose. Uh, but you can't get that stuff out of your house even. The pollen's so small. So it's pretty amazing what can be accomplished with wind. And some pollen is quite heavy and relies on insects or even animals to be spread. Um, this is just a fun picture I grabbed off the net that just shows specific pollinators for specific flowers. I mean, there's hundreds of thousands of associations. Some of them are incredibly specific. Some flowers are very generic. They just don't care as long as something lands on them. But some are very, very specific. You could spend ages talking about that, but it's beyond the scope of this course. Um, in sexual reproduction, obviously we've got pollen, we've got flowers, we have compatibility. The next step is fertilization. So this is the process where the pollen lands on the stigma, and then the pollen actually absorbs water and begins to grow a pollen tube down in through the stigma into the style and all the way down around into the ovary, ultimately delivering genetic information to the egg cell, in this case a sperm cell or sperm nucleus, to the egg cell or egg nucleus, and then forming an embryo. This is a hyperlink here that I would highly recommend you watch. It's in the PowerPoint version of this. It's a great visual explanation for how double fertilization happens. So let's look a little deeper. So pollen takes up water when it contacts a stigma, germinates a pollen tube that travels down the style. And as it's traveling, it's got a tube nucleus and it has basically a sperm nucleus or a sperm cell. And as it progresses, that sperm cell actually divides into two. So you can see in this picture, uh, there's actually one, two sperm nuclei, and then there's a tube nucleus at the front. So there's three of them on their way. That typically happens closer to where it's going to enter the ovary. So the pollen tube grows down through the stigma style in what's called the transmitting tissue. That's a specialized tissue that helps guide the tubes. Otherwise, they don't really know where they're going, down. How do they know where to go? So these tissues help guide the pollen tube. And while I'm here, I might as well mention that this is one of the ways that plants can sort of test the genetic strength of the pollen that's landing on them. Oftentimes, the distance that pollen tube has to grow is significant. And if pollen's coming from a weak plant, it may not have enough energy for the pollen tube to make it all the way to the ovary. So by having a convoluted pathway, it's testing the strength of the pollen to make sure that whichever one is successful at germinating the egg cell is most likely to have a strength in or some adaptation that might be advantageous, as opposed to allowing a weak germ cell to our sperm cell to fertilize. So as the tube is arriving, the germ or the sperm cell divides into two, right? And these two cell things are traveling along. Here's a little picture to show you. Just threw this in to give you an idea of what we call chemotaxis. This is where a pollen tube is actually following a piece of an ovary that's been cut out under a microscope in real time. So you can see how it quickly it moves. Pretty wild. And the image on the left is a fluorescence dye that's been added to a stigma showing all the pollen that have germinated on there. And many times there's lots of area there for pollen to germinate on top of, but the distance is such that the pollen tubes have to make a long ways to the ovary. So that's a good example of where having a distance to test which ones can make it is advantageous to that flower. So as the pollen tube approaches what we call the micropyle, that's the opening to the ovary, uh, the germ cell divides into two sperm nuclei. All right, so remember how I mentioned there would be three ultimately. So we end up with these two haploid sperm nuclei. They have half the normal amount of DNA. They progress into the ovary, and one fuses with this egg cell, forming a zygote that's down here. And the other travels up to the center where there is two haploid cells waiting, and it forms a triploid endosperm. So that's probably the key information out of what a double fertilization is, in the sense that we're forming a zygote and we're forming a triploid. Fancy terms here, synergids. These are the cells that are on either side of the egg cell, and they are guardians of the entrance, the micropyle. So the tube has to pass through them, and they will permit that as one last check, kind of think of it like that. And where the pollen tube goes, that synergid disintegrates and opens a pathway, and the tube enters, delivers the two nuclei, and away you go. And here we have the triploid endosperm. So this becomes the cotyledon. So that's where all the energy is contained to drive the growth of the embryo when you germinate it. You might say, why triploid? What's with all that? Well, part of it is that this is a very large structure, ultimately, that's governed by these nuclei, and the extra DNA helps to code for enough of the protein to develop this complex structure to feed the embryo. Again, that's beyond the scope of this course, so we'll just move on. So double fertilization, right? Primary endosperm divides, forming the endosperm, and then we got your zygote as well, which is ultimately the embryo. And here, I'm just looking at a sweet pea. I think it's kind of fun to look at this. The whole pea pod here is the ovary of the flower. And the petals would have been on this end, right? And the ovaries in the middle here, as you can see on this picture. And what's happened is the pollen have germinated, and they have traveled along the style into each individual ovary and fertilized. Now, one pollen per ovary, obviously. Each one has been fertilized. Now, you'll notice this one didn't get fertilized very well, so that's a bit of a dud. Here's a dud down here. There may have been something wrong with the genetics in those particular pollen. But so that's the cross-section of the pea ovary, and you can see each pea that we see, the kind maybe you don't like peas. I love them, but each one of these was a separate pollination event. And just like a corn cob, all the hairs that are in that corn cob, remember the tassels? Pollen lands on a tassel, and each tassel is guiding a pollen tube a long distance to each corn kernel. And if you think about it, given the size of a corn cob, that's a long distance for the pollen to grow from a tassel all the way down and long into the ovary to fertilize each kernel. And as a result, corn pollen is actually quite big because it has to have a lot of energy in there to drive the growth of that great big long pollen tube. So anyways, that's double fertilization. I threw this slide in just for fun, talking about spreading seeds. This is jewel weed, and it's spread by mechanical contact. I always have fun with these in the summer. Um, they like to grow in wet places, often near poison ivy. Their sap is purported to reduce the itchiness of poison ivy if you get it. So I don't know how true that is, but anyways, let's moving on. Moving over to vegetative propagation. This is the process of essentially reproducing a plant without going through a fertilization event, right? So we've all taken cuttings and stuck them in a jar. No, that's not rocket science. Lots of us have probably done that. That is asexual propagation. The plants that result are genetically clones, essentially. Sometimes you can get variation happening depending on how much stress the plant it goes through in the propagation process. But generally, we're talking about the terms would be clones, propagules, plugs, any kind of young juvenile plant. The result that's derived from the result of taking cuttings and rooting them out. Kalanchoe, some fun ones with little propagules on the ends of the leaves. So typically we have three options. You have vegetative tissue cutting, so these are non-woody, in this case chrysanthemums. Semi-hardwood cuttings, in this case rosemary. And I'll let you guess what those are. Hardwood cuttings, they've got secondary wood and bark. They each have their own strategies, but it is interesting to note that they do vary. The processes can vary depending on what you're starting with. So cuttings have two mechanisms for rooting, right? So we have what's called adventitious roots, or we have morphogenic roots. In adventitious roots, these are preformed on the plant and they sort of lay dormant. And when they come into contact with soil in the right conditions, like moist, they will begin to grow and infiltrate the soil and establish the plant. Where morphogenic roots, this is where cells have to reorganize and lay down a foundation by growing new cells through what we call callus. So let's look at adventitious roots first. So on this is English ivy here, you can see that there's we call root primordia sitting on the stem waiting for when that touches the soil. And so yes, a lot of vines propagate like this. Think about it from a perspective when they're out in their natural climate. Vines are creeping along the ground and they are trying to take over the world, and so they will root out wherever they touch. So whenever they touch the ground, they root out. And if a plant, animal, or whatever happens to cut it, it doesn't matter. You now have two, and they just continue growing from here, and now you have more vines. And so they tend to take over the entire area. Of course, I've already mentioned it, but poison ivy is just a horrendous example of plants that will propagate and take over the world, technically from under the ground, so not advantageous. But anyways, vines, very common. Here's a close-up picture of an adventitious root that's just laying below the surface, just ready to come out. As far as propagating, adventitious roots allow us to do what's called layering. You'll probably run into this in some other courses, but that's okay. We'll introduce it now. Layering essentially means that if you take a tissue, and in this case you bend it down and bury it in the ground, it will start to form roots adventitiously from the sides of the stem. Quite a few woody perennials will propagate like this. You can also stick the ends directly in the ground, blind as we call it. They'll propagate. Snaking, lots of things. You can mound. This happens sometimes when people mound their roses and forget to pull the soil away in the spring, and they end up with lots of stems rooting out. The one on the left here, down here, this is probably a little more extreme. Uh, you basically pin the plant down and bury it in the ground, and then it shoots up. I don't know. That seems like medieval torture propagation. um, Layering is often employed by just putting a stone right over a stem on the ground. Down here in the bottom, this is a blueberry. Yeah, it's sitting dormant right now. And the branches are being held onto the soil by a stone. It exits the base and comes up over here. This one comes up over here from the base. And eventually, they'll root out under the stones, and then you can remove the stones, cut those branches off, and put them in another part of your garden. So, very common for propagating blueberries. Now, what do you do when there's no root primordia or adventitious roots? So let's say you take a nice vegetative cutting, there's no roots on it. What are we going to do? What happens? All right. Um, let's review a couple of things from the tissue culture class and talk about that briefly. So totipotency is a term that is critical for propagation. That means that every single plant cell has the information and the capability of producing an entirely new plant. And we rely on that for tissue culture to work, right? We're going to take an X plant and we're going to put it through tissue culture. We need totipotency in order for that to form a fully formed plant. So typically we see lots of growth in meristems, apical, lateral meristems, root meristems, lots of dividing cells and new tissues being formed. But what does this have to do with propagation? Well, when you wound tissues, they revert to meristematic activity. So you've probably seen a branch has been cut off a tree and you get this sort of lumpy growth around it. That's a wound that's healed. And that reorganized itself and filled the gaps, sealed it off, formed bark, began to organize its growth, and basically covered over that area. So wounding is a big part of propagation. So plants form callus in response to wounds, right? And callus are undifferentiated cells. And here's the tissue culture kind of gnarly little callus chunks that haven't yet received enough of a hormone signal to tell it to become a root or a shoot. So you just get this sort of like almost a cancerous growth kind of. That's callus. Um, Once the callus begins to organize itself in the presence of plant hormones, it will begin to form proper roots and shoots. In this case over here, this cutting has been dipped in a rooting compound, stuck into some soil, and eventually it forms new plantlets. All right. Uh, you can rely on what we call exogenous applications of plant hormone or endogenous, which means the hormones that are in the plant all along. Every species is different and they require different amounts of hormone. You'll learn more about that as you go through the program, but there's different strengths of rooting compound, and that's related to how responsive the tissue is. And a lot of times plants don't even need it. And ironically, African violets are a really good example of a cutting that doesn't require exogenous plant hormones, but in this picture, they already did it. Um, if we take a closer look at what's happening with those cuttings, you'll see that there's kind of swollen, undifferentiated growth. You can see that on the end over here. And the tissues are starting to organize themselves and grow roots. And as they grow more and more roots, there's less callus produced and just energy goes into root formation. So we're always looking for callus to assess the success of rooting. One of the tricks as a grower when you're assessing your cuttings is you will check your bench, you'll pull the cutting out and you slide your two fingers along the stem towards the bottom. And if your finger slides straight off the end without any kind of change in thickness at the bottom, you know you're still at the early days and you need to, and if it's been a long time already, you need to change something, maybe more heat or whatever. But if you feel a bump at the bottom, then you know that your cuttings are responding and that things are moving along just nicely. Other forms of propagation, which we've covered, tissue culture we've already talked about, wounding I've talked about. I should highlight because this air layering often is associated with layering because it's kind of practiced in the same group of activities. But technically, air layering involves wounding because you're cutting the bark back, which triggers meristematic activity where the wounds are in the bark. And once they go through a callus phase, they start to produce roots into this soil. And what typically happens is you you damage the bark, and then you take a plastic tube or a sheet of plastic, tie it around the bottom part of the stem, and then fill it with soil and tie the top. And that soil should be damp. And then that damp soil that's in direct contact with the stem, as the plant forms callus, they will directly root into that bag. And you can inspect the bag periodically. And when you see there's enough roots, you open up the bag, wash it off, cut the stem off, and then transplant it. And you've made yourself another fruit tree or whatever it is you're trying to do. And then another one, which I don't want to spend a ton of time on because you're going to cover some of this with Mary Jane, is grafting. Now, grafting is a really interesting process. It's an art as much as a science to do it well. But it is definitely an interesting way to propagate. Is it a way to propagate in vast quantities? Not really. It often is employed more for other reasons. When you graft, it increases nutrient uptake. Maybe you have a better or stronger root system. It can make your plants more hardy. Most of the grapes grown in Niagara that are for wine are grafted, French varieties that are grafted onto rootstock that is winter hardy. And sometimes it's not winter hardy enough because our cold winters, they do occasionally die. But if we left the roots on these French varieties that were there originally and planted them here, they would probably die. The other thing is that you can impart disease or viral resistance with grafting. So sometimes rootstock will actually impart disease resistance to the top part, which we call the scion, the part that we're grafting on. In this picture, you can see the stems have been jammed into a wedge. I think for plants that are fairly easy to graft, that might work. That would be tricky to keep it clean and stuff. You usually have to put a lot of wax or something to keep that wound clean. Grafting has also been used extensively in the greenhouse industry. Pretty much all tomato and pepper plants grown in greenhouses are grafted with a scion stalk that produces desirable fruit onto a higher performance rootstock that can really push the fruit to increase yields and also impart disease resistance. There's also a variety it's actually a cultivar because it's propagated through non-sexual means. Mighty Mato is a grafted product that was marketed to garden centers for a few years. And it never really took off because obviously if you're going through the process of grafting, there's added cost. And I don't think people really understood the difference or what was going on. And the company that was marketing that just never really did all right. I think they still sell them, but it never really took off. But yes, they produce more tomatoes for a given plant. Obviously, you got to feed it to match it. It's not going to produce more tomatoes from thin air. So you got to know how to feed it and follow the instructions. Anyways, if you see a Mighty Mato in the garden center next year and you really want to fork out the extra cash, give it a try. Let me know. A key thing here, matching stem thickness for success in grafting, especially if you're doing butt grafting. The vasculature, the closer the two vasculatures are to each other, the more likely you are to succeed in your grafting because it takes less time for the phloem to flow into the new phloem and the xylem, etc. So it's usually helpful if the stems are similarly matched. And if you're mismatching sizes, at least make sure that part of the vascular cambium is overlapping to improve your success. The other thing that's helpful is that the general health of the plants is good, so that they have enough energy and vigor in them to push through that time where the grafts are taking the energy to grow together. Because each half, the scion and the rootstock, will have to feed on their own energy reserves until those connections are made. So healthy, strong plants are required. Further plant adaptations for propagation are what we call fragmentation or stolons, structural adaptations like stolons, rhizomes, corms, bulbs, and tubers. So um, I showed you a picture earlier of a calancho, and that's technically fragmentation. That's where the plant produces propagules, or basically miniature plants that are fully formed. And they can fall off onto the ground and continue growing happily, producing lots of young. Um, Not that many plants are able to do that, but it's pretty cool, so we bring it up. Now, stolons, or sometimes referred to as runners, are a stem adaptation that grows above the ground. And just like in a strawberry, it will touch the ground, and where it touches, it will root out, producing another young plant. Mints do this as well, as they try to take over the ditch around your house. Uh, Typically, plants that grow via stolons, or even the next one, rhizomes, are aggressive and can take over an area real quickly. Um, Rhizomes are basically an underground version of a stolon. Uh, if you look at all the phragmites growing in the ponds at the corner of the entrance to the campus, you'll see just how aggressive these rhizomes can be. Um, Giant reedgrass, a lot of big grasses produce rhizomes. And then the ones you might be more familiar with in the kitchen, ginger. You can go buy that at the grocery store, and you can actually grow that if you want. You can grow your own ginger. Those are rhizome. They also have starch storage, so they have energy reserves, and that's why they're sort of thick, depending on the structure. Um, and in the ornamental world, irises grow from rhizomes. Another adaptation are corms, gladiolas, freesia, cyclamen. These grow from corms, and they typically have what's called a modified stem basal plate. That's a fancy word, but on this picture we have the stem, and then the bottom of the stem swells, and at the bottom you'll see there's roots coming off a section. We call that the basal plate. So the corm, that's the basal plate down here. The corm is a structure that will form cormlets off to the sides. You'll see these little cormlets forming, and they will begin to grow and form daughter plants or clones of the original plant. In some cases, you can even cut the corm. You can divide it. Typically, you want to include the basal plate, some of it in each half, if you want to be successful. But you got to be really careful that once you cut this open, bacteria and fungi can get in and rot out each piece. So commercially, they often will treat with any kind of a fungicide or bactericide on these before they plant them. Then there's bulbs. You're familiar with onions. They're a bulb, which is a compressed stem. So the stem for the entire plant is kind of in this area here. And then these leaves come up and form long tubes, of which your leaves and your flowers form. So bulbs produce bulblets, which are just like the corm with cormlets. The bulblets grow around the bottom. In this picture up here, you can see there's bulblets that have matured and formed plants neighboring to probably the original one is here. It's an interesting strategy because you can definitely crowd out competition, but your offspring can also crowd out your own roots. So hopefully the ground can support all that growth. And the starches are in the leaves. Like that's why onions taste so good, because they're full of starch. When you cook them, they start to sweeten. The onions add a lot of sugar to food when you cook them. Oops, that's supposed to be an eraser. Nope, not working. Anyways, so bulbs are another means of propagation. Here's a popular one, tubers, your standard potato. And in the ornamental world, the caladium is an example of a tuber type plant. And here we have a potato where the eyes are sprouting, right? So you can see it's forming new growth, being fueled by starch that's stored inside the potato. Typically, you want to include at least one upright eye in your piece of potato. So if you're cutting this up, you want to propagate it by slicing the tuber up. That's fine. You can do that. Again, be mindful that where there's wounds, bacteria and fungus can get in and rot. But there has to be at least one eye, this is an eye, facing upright for it to work. The main reason for that is that it takes energy for the plant to push through growth. And if you've removed a lot of the tuber, there's less starch reserve for that plant. And if it's upside down, the shoot has to grow around and up and then finally make it to the top of the soil where it can begin to form leaves and undergo chlorophyll, like here. And if it's already small and you've made it that much harder for it to germinate and grow up to the soil surface, you reduce your chances of success. Anyways, so typically young potato pieces are cut with several eyes so that when you throw them in, the odds are one is closer to the surface. And unless you're carefully placing everything. Well, I hope that you've learned something about propagation. I haven't gone into the more of the growing side of it because you're going to get that in Mary Jane's courses and other courses. So I thought I'd talk mostly about propagation structures. All right. Next week is review, and then you're off to your exams. I hope you've enjoyed the term, and I hope you've learned something interesting. All right. Take care.